Hello, everyone. Hope you're doing well. First of all, I'd like to thank you, of course, for staying till this time. I know it's a bit late now, but for giving me the time to speak. I'd like to thank the scientific committee for giving me the opportunity to talk about this rare disorder and the sponsors as well. So I know it's late, I'll keep it brief and let's go. So, I'll be talking about neurodegeneration with brain iron accumulation 5 or NIBIA 5, also called BPAN. This presentation was prepared by myself and Dr. Ahmed Shatila. First of all, I'll talk about the case and then we'll dive into the NIBIA word, right? So, patient history. So, a female patient with a birth injury, lack of oxygen due to the wrapping of the cord around the neck. She was delivered through a lower segment cesarean section. So, following birth, cried normally, was not admitted to ICU or any type of care, urgent care. Uh, she had delayed walking. She started walking around 2 years old. This is according to the family, of course, and no problems related to walking whatsoever until she became 9 years old. She started using a wheelchair. If she wanna go, if the family is going out, so they just use a wheelchair. In the house, she walks on her knees. This happened due to sudden increased stiffness and reduced mobility. So, according to the family as well, she never talked. And later on, she was diagnosed with spastic cerebral palsy and long-standing epilepsy. So this diagnosis came from multiple centers in the UAE and Europe. So what were the observations? I'll just go through them quickly. She had high BP, increased CK levels, motor disability, insomnia, seizures, anorexia, anemia, asthma, hernia. As you can see, it's a long list, and I did not mention all of the symptoms or manifestations, but these are the main ones. So, as I told you, the diagnosis was agreed on by multiple physicians. So, they only prescribed her symptomatic treatments, such as iron supplements, vitamins, Nexium, baclofen, and so on. So, in 2022, February 2022, she had a neuro consult with Dr. Ahmed Shatila, and he requested a whole genome sequencing and mitochondrial genome tests. The results came back positive for a mutation in the WDR45 gene, which is associated with NIBIA5, which is also called as well BPAN. So, let's dive a bit into NIBIA. But let's talk about first, because NIBIA is related to iron, so let's talk about iron a bit and then we'll go into NIBIA. So the brain or the human brain relies heavily on iron since it is involved in multiple processes such as DNA synthesis and repair, neurogenesis, axon myelination, neurotransmitter synthesis, energy production, phospholipid metabolism, and so on. But iron levels in the brain, they're different throughout life stages. So at birth, not detectable iron levels. Iron starts accumulating throughout life or throughout development of the human, let's say. And so the levels of iron are different among different cell types. So high concentrations are found in oligodendrocytes, in the cortex, and cortico-subcortical junctions, in addition to areas of myelination. On the other hand, low levels of iron were found in astrocytes and in neurons. So I can't see because the recording is covering a bit of the slide, but the idea is during adulthood and old age, the accumulation of iron starts to increase in specific cells. For example, in astrocytes, microglia, and where else? Red nucleus, putamen. But the iron levels go down in the frontal gray matter, thalamus, and caudate. Now, since we talked a bit about iron, the functions, levels, now let's talk about nibia or neurodegeneration with brain iron accumulation. So basically, nibia is a heterogeneous group of inherited diseases caused by excessive accumulation of iron depositions in the brain, mainly in the basal ganglia and substantia nigra. For now, we only have identified 10 genetic mutations that corresponds to 10 different subtypes of nibia. Majority of them have autosomal recessive inheritance pattern, except for nibia 3, which is autosomal dominant, and nibia 5, which is BPAN as well. It has a X-linked dominant inheritance pattern. So nibia 4 mainly has autosomal recessive pattern, but sometimes occurs with autosomal dominant pattern. So the disease course could either could have a slow or rapid progression due to the accumulation of iron, but most reports indicate that it's always a rapid progression. But some of them mention that it could be slow and maintained for a while. So the nibia disorders in general, they exhibit a complex multi-system dysfunction which is induced by the progressive neurodegeneration, and where the extrapyramidal symptoms such as dystonia mainly, dysphagia, spasticity, ocular motor dysfunction, vision loss, like among of course other symptoms that they have been noted in multiple reports. And there are other core symptoms in nibia. These include these symptoms or core symptoms can be seen in all nibia subtypes. Patients could have a decline in their psychiatric and cognitive status. Usually when all these symptoms combine, they lead to early death. Okay, so this is a huge table, I know, but I'll just go through it quickly, won't mention everything. So, as I mentioned before, each gene mutation corresponds to a different nibia subtype, right? So let's say nibia 5, BPAN. The gene responsible is WDR45, located on chromosome X. The features of this mutation or of this disorder, I don't know if you can see it, but if you can see my mouse, I'm not sure, but it disrupts this mutation disrupts autophagy, causes impairments in elimination of abnormal and toxic proteins, and causes cellular stress, which leads eventually to death. And then I mentioned before that 10 mutations correspond to different nibia subtypes, except for idiopathic nibia. No gene mutation has been identified. Nothing has been identified. Diagnosing idiopathic nibia is based on having all the features or characteristics of nibia, but with negative results of any of the nibia genes. Again, so there are multiple groupings of these disorders or gene mutations. So let's go through them briefly. So these mutations in these genes leads to impaired myelination, myelin maintenance, and phospholipid synthesis. In the WDR45 and ATP13A2, impairment in autophagosome formation and clearance of iron. FLT leads to dysfunction in leads to failure in recruiting excessive iron levels, and CP leads to failure of exporting iron outside the cell. Another grouping, so with nibia disorders having heterogeneous pathological mechanisms, they can be regrouped into three main categories. First one is induced by proteins involved in lipid membrane homeostasis and lipid metabolism, induced by proteins involved in iron metabolism, and other forms, which is nibia 5, idiopathic, and then you can see NIBIA3 and proteins involved in iron metabolism. And let's say, okay, NIBIA4, the MPAN, which is related to the mitochondria, falls under the group of induced proteins involved in lipid membrane hemostasis and lipid metabolism. In general, they are not age specific and affect both males and females equally, except for NIBIA5 occurs more in females. So all NIBIAs, they fall under the category of ultra-rare neurodegenerative disorders, where the incidence rate is 1 to 3 individuals per million, of course, according to the NIBIA Disorders Association and other published literature. So diagnosing any of the NIBIA disorders would be a bit challenging because the symptoms or clinical presentation could overlap between either NIBIA subtypes or between NIBIA and other disorders such as leukodystrophies. So diagnosing NIBIA basically depends on 3 steps. First one being identifying the phenotypes of NIBIA. Second one, brain MRI using iron sensitive sequences. So mainly that the abnormalities are seen in the central nervous system, brain, but rarely could be seen in the peripheral nervous system. And eventually molecular genetic testing, for example, whole genome sequencing. It is important to obtain a 3-generation family history to identify any NIBIA manifestations in the relatives. So let's talk about the imaging characteristics of 4 major subtypes of NIBIA. So for the sake of time, I'll just talk about one because I know it's a bit late. So the first one, A, PACAN. So using a T2 sequence, what can we see? GP hypointensity indicating iron accumulation with an anterior medially located area of hyperintensity, which is also known as the eye of the tiger. In picture B, we can see hypointensity in the medial aspect of the nucleus. Now, later on, we'll talk about a different type, so that's why I'll just go to the next slide. Now, NIBIA, BPAN. NIBIA or BPAN is a neurodegenerative disorder caused by a mutation in the WDR45 gene, which is found on chromosome X, chromosomal location P11.23. So generally, all diagnosed patients with BPAN shows de novo mutations in the WDR45 gene, and with having clinical features that do not usually follow an X-linked disorder pattern. Initially, BPAN was referred to as static encephalopathy with childhood with neurodegeneration in adulthood, but later on was established as a form of NIBIA. So BPAN affects both males and females, but with the tendency to affect females more, which could be explained by the following points. BPAN in males is thought to be caused by somatic mosaicism through postzygotic mutations. In females, they can carry either a germline or somatic mutations, which could possibly be associated with the X chromosome skewed methylation pattern. Now, in males, it could be lethal before birth because the majority of the WDR45 mutations are de novo, which could lead eventually to early death. So, as mutations occur in the WDR45, the protein coding function is lost, which eventually leads to impairment in autophagy, which is the hallmark of BPAN. To date, we don't know the exact pathophysiology of BPAN, but we know that the WDR45 protein is a member of the WD40 protein family, right? And this family has a beta-propeller platform structure. They are involved in multiple cellular functions, such as signal transduction, transcriptional regulation, and autophagy, which is particularly seen in WDR45 protein in regulating autophagosome formation. So Some investigators have coupled both fibroblasts from a female diagnosed with BPAN with iPSCs, derived midbrain neurons, to try and understand what actually happens in BPAN. So they noticed that there are increased levels of intracellular iron, decreased levels of LH and mitochondrial ferritin, increased oxidative stress, defects in autophagy, and reduced lysosomal function. Now, when they increased the levels of WDR45 protein, all abnormalities went back to normal. Now, in other studies, some investigators have reported tau positive neurofibrillary tangles in the hippocampus. So it's not only about iron anymore. Now tau is involved, of course, due to the neurodegeneration. But from what we can get from this slide, it's a multifactorial disease. So BPAN usually has two stages for the disease course. First one, global developmental delay during childhood in addition to intellectual disability. In stage 2, progressive dementia, dystonia. Now, the idea is as patients get older, symptoms start to add up, of course, due to the progressive nature of the disease. Now, later on, patients start to suffer from shortened sleep onset latency, problems with REM sleep, and of course, again, due to the rapid progression of the disease course. Now, multiple reports have documented a Rett syndrome-like phenotype with repetitive midline hand wringing in BPAN patients. When patients get into advanced stages of the disease, they start suffering from unstable behavior and become bedbound. Now, other ocular abnormalities have been reported in multiple studies with patients diagnosed with BPAN. Now, as a common symptom between all BPAN patients, seizures. They usually start in early infancy and early childhood, which is likely to stop during adolescence. So other types of seizures also reported. Generalized seizures, focal seizures with impaired consciousness, and epileptic spasms. Now, the pattern of seizures in BPAN patients could be similar to other epileptic syndromes, such as West syndrome, usually worse early during childhood and lessen with age. Now, multiple brain MRI findings have been noted in BPAN patients. First one, iron accumulation in both SN and GP during early stages of the disease. Hypomyelation in thin corpus callosum during early childhood. T1 hyperintense halo signal, in addition to central band hypointensity in the SN. Now, the halo sign or the halo signal is specific to BPAN. So as you can see in the picture, this is the hallmark, or the halo sign is specific to BPAN. That's it. And of course, brain atrophy, which could occur at any age. So in conclusion, for now, no curative treatments or disease-modifying therapies are available for NIBIA 5 or any of the NIBIA disorders. Patients are only offered symptomatic treatments. Multiple investigators around the globe are putting their efforts into applying research methods and designs in BPAN-related studies. Even with all the conducted studies, we still need more research to understand the exact pathophysiology of the disease, in addition to providing patients with a cure rather than symptomatic treatments. Now, these are some of the published articles or studies related to BPAN. Of course, there are a lot more. So if you're interested, just reach out. I'll send you the link. You can go through all of them at once. And thank you.